Dr. Paper. This is Danielle, Rumsey.
Audrey.
And Jedzia.
And our gene is the ataxin-1 gene.
All right, so some background information for the ataxin-1 gene is that its function is that it encodes the DNA binding protein ataxin-1. It is a polyglutamine expansion within ataxin-1 that is involved in transcriptional repression. The causative gene of spinocerebellar ataxia type 1, SCA1, is a neurodegenerative disorder, and ataxin-1 triggers formations of NIs. There we go. So for research and discovery, inheritance and mapping, the ataxin-1 gene was discovered in a lab by Dr. Huda Zagabi and Dr. Harry Orr at University of Minnesota. They discovered that mutations that repeat a region of the gene led to SCA1, which, as further discussed in the background info, leads to neurodegenerative disorder. For inheritance, ataxin-1 is a autosomal dominantly inherited neurodegenerative disorder that is caused by the repeat of the trinucleotide that codes for glutamine in the protein ataxin-1. Through generations, the mutated gene is passed down and increases in length, making the protein more unstable. For mapping is a picture of the locus, chromosome 6 in the right, and you can see ataxin-1 on the left of the chromosome 6. For molecular function, SCA is genetic abnormalities is most commonly caused by a CAG repeat expansion, leading to a polyglutamine expanded protein product. This is actually toxic to the neurons, and there are multiple variants of SCA1, which is due to alternate splicing. Alternative splicing causes an overlap in alternative reading frames, which then results in multiple transcript variants, each of which encode many different proteins. The normal ataxin-1, the CAG is repeated like 6 to 36 times, while the mutant form repeats more than 100 times. This expansion is anticipated, and the cones faster and more severe in future generations, leading to more harm. Ataxin-1 is localized in the nucleus of neuronal cells and cytoplasm. The mutant has a role in transcription repressing, seen in the next slide.
Okay, so for the first primary lit that we looked at, just for some background information, they said in their introduction, the poly-Q expansion causes the ability to bind to RNA to decrease, and this is thought to be due to errors in the DNA machinery. The poly-Q ataxin-1 aggregates, and they form inclusions in the nucleus and cytoplasm, and this occurs mostly in Purkinje cells in the brain. And because it forms these aggregates and its ability to bind to DNA to decrease, it alters cellular pathways, obviously transcription and RNA processing. It also alters signal transduction, leading to cell death. So basically in this experiment, they wanted to see what the source of symptoms is in SCA1. So they wanted to see if it was just due to the aggregates and inclusions in Purkinje cells, or if it was due to other effects. So what they did, and this was in the early stages of the discovery, so they didn't really know the cause of SCA1 yet, or what caused the symptoms. So basically what they did was they generated 2 mouse models. So they first created a BO5 transgenic mouse model. So they overexpress a poly-Q ataxin gene. And doing this, they just knocked out the Purkinje cells to be present. And so basically they just saw the neurological phenotype of SCA1. And they did this by using the human SCA1 from cDNA. So they used the mRNA to get the DNA from it. And then they expressed it through the promoter of the PCP2 and SCA1 transgene. The second mouse model they did was they created an SCA1 knock-in model, which they had 154 CAG expansion into the exon 8 of the endogenous ataxin-1 locus. So they expanded the CAG repeats, making the SCA1 phenotype be expressed. So they wanted to see if the phenotype was just the same in both models with the Purkinje cell expression turned off, or if it also had different expressions from having the polyQ in the cortex, thalamus, hippocampus, caudate nucleus, putamen, brainstem, cerebellum, and spinal cord. So their figure 2 just showed the different symptoms they had between their mouse models. And so basically they saw that the transgenic mice had no cognitive impairment, but it had motor and coordination. And then the SCA1 phenotype showed a lot more. So they had motor and coordination, muscle wasting, cognitive impairment, and memory deficits. And so they saw that because of this, the phenotype of SCA1 is not just due to the Purkinje cells, but also due to everything else that I listed before. So for the second primary literature experiment, they wanted to see how the wild type of ataxin-1 reactive proteins, because they only knew the function due to the SCA1 phenotype from the mutant form. And so they wanted to see its interaction with the transcriptional co-repressor SMARTR. And SMARTR is a silence mediator of thyroid and retinoid hormone receptors. And so they wanted to see if they were linked and if they interact with each other. So the methods for this is they took the human wild type ataxin gene and they isolated it from the cerebellum poly-A RNA library. And they did this using PCR, so they amplified it. And then in order to express the gene, they used 2 primers for the ATXN1 gene. And what they did was they had a CGA repeat deletion generated, done by 3-way ligation. And so they did, they wanted to see if the CAG repeat had a different interaction with SMARTR. So what they did was they altered the deletion to having 0Q or 30Q or 82Q. In the taxin gene. So they just changed the amount of CAG repeats to see if it changed the interaction with SMARTR. And so they did this by, they took the human ataxin-1 amino acids generated by the PCR, and then they subcloned them into an expression vector to see how the protein interacted with SMARTR. And they did this using direct link yeast 2 hybridasase and co-immunoprecipitation studies. So in figure 2, you can see that they used beta-galactosidase, and this showed the detection of the expression of the recombinant genes. So you can see and see how they lit up the areas of SMARTR and ataxin-1 and how they were basically in the same area. And then in D, you can see the activity of beta-galactosidase in the 0Q, 30Q, and 82Q. And so what you can see is that it didn't really change the activity, showing that the glutamine repeat doesn't ruin the interaction between 2 proteins, but the function of it was still unknown. So if it doesn't ruin the interaction, what difference does the poly-Q tail have in the 82Q compared to the 0 and 30Q? So they saw in a previous experiment that ataxyl in Drosophila acts with the SMARTR in vitro, and so they wanted to see how it reacted in Drosophila tissue. And so they examined whether the 82Q forms aggregates in the salivary glands of Drosophila and whether the SMARTR's expression is affected from it. And so they did immunostaining of the salivary glands with both the Ataxel and SMARTer specific antibodies. And so they basically washed it with antibodies to light up the areas where those genes are present. So what they saw was in A, B, and C, or I guess A through E, they saw the aggregates. And C, you can really see how it forms the aggregates in salivary glands. So the Ataxel 82Q binds to the chromosomes and then sequesters SMARTer into aggregates. And then you can see on F that they lit up the areas of the chromosome with Ataxin 1 and then F with SMARTer. And they merged these two photos in the bottom F, the F prime, I guess, the yellow one. And they showed that they were both, they're co-localized each other on the polytene chromosomes. Polytene meaning like they have high areas of transcription. And therefore, the transcription co-repressor SMARTer, explained by the polytene areas and deformities, shows that the Ataxel is a chromatin binding factor and is likely involved in gene transcriptional regulation from seeing its interaction with SMARTer. So, yeah, pretty cool.
All right, so to continue our story, the next study they did was on BOAT, which is, they call it brother of Axin 1, and it shares a specific protein. They wanted to see if it would share specific, this AXH protein domain that both Axin 1 and BOAT need to interact with SMARTer, the one that we just talked about. And so they wanted to investigate the potentially interconnected relationships between Ataxin 1, BOAT, and SMARTer proteins. So they did this through using BOAT cDNA fragments that were isolated from a human cerebellum poly-A RNA library, and they did this through PCR. And they made many different fusion constructs by subcloning the fragments into different vectors, and then they generated antibodies for BOAT, Axin 1, and SMARTer. All right, so figure 3 shows the properties of BOAT and Axin 1. They found that they were very similar, and that the AXH domain-dependent interaction between BOAT and SMARTer, N-core R and SMARTer, their interactions were also found between BOAT and SMARTer for different plasmids. And then in figure 4, the AXH protein domain for BOAT and Axin 1, they found this overlap, and you can see in the bottom in F, it shows where BOAT overlaps with Axin 1 in this 469 to 582 for BOAT, and then in Axin 1, 575 to 688. So what they discovered through this experiment was that the Axin 1 associates with BOAT and SMART and SMARTer transcriptional core pressors through this conserved AXH domain. And they succeeded in finding a novel human AXH domain protein, which is BOAT, and SMARTer's interaction confirmed through human cultured cells. So close properties of Axin 1 and BOAT were also discovered, which means that the proteins likely have overlapping transcriptional regulatory pathways. BOAT interacts directly with Axin 1, and its cytotoxicity was suppressed, so that has really good benefits for the future studies as well. And then the AXH domain structure was also found, and that was just kind of fun that they discovered. So what does this gene product interact with? Axin 1 binds to RNA and is associated with many proteins in large complexes, one of those being discovered like the AXH domain. It has been discovered working in transcriptional repression that regulated Notch, which is signaling pathways, and Capucha, which are protein and regulates gene transcription.
All right, so for this one, there were like 3 purposes that are the big keys what they did in this. The first was determine if the chromosome deletion in patient 1 who has ASD was the chromosome deleted part 6p22.3 through p24.3 was the cause, and if this is the chromosome that was also affecting the ASD or creating ASD. How they did this was that they compared patient 1 using a array CGH to 5 other patients who also had ASD, and they were also had the chromosome deletion part. That was the first part, which, if you look at the second slide, I lost my slide. Right here on figure A shows you patient 1, and you can tell that that's how much was deleted out. And the further out you get, like for patient 3, since she has a chunk missing, the more palms she's going to have, which was shown throughout the study. We go back to the first page, please. The second part was to figure out whether to understand the origin of the deletion. They did this using an SMP analysis on patient 1 that showed that all 1,193 patients that the SMP had discovered showed that the deleted region was found on the mom, but it was the same as the normal chromosome. So there was no big difference. But it also shows that it was inherited from the mother or whoever had this, that she was not a carrier though. The third part of the study was that they were deciding if ataxin was the cause for the ASD that occurred, or if it was multiple genes functioning together. And they had 16 patients they found that linked up with patient 1, and out of 13 of them, they had ataxin 1 that was deleted. So it wasn't all of them that had it, so this also indicates that other factors, like other genes, might have been needed to help control or, change it. And that the amount of the gene in the specific reason had an important cause. For this, the methods that they used were, for the very first one, was the hybridization array of CGH, which was just comparing patient 1 to the other patients using a CAG repeat polymorphin in ataxin 1. And then for the associating where the origin of it came from was using an SMP array analysis. Going on to the last slide. This is basically what they discovered. I totally forgot about this slide.
So what did they discover, Jed?
They discovered that the deletion found in the chromosome did result in ASD, but locating which gene in the chromosome is important, because it turns out that not just ataxin 1 is causing ASD. And they also found that patients 2 through 6 had 16 deletions that overlapped with deletion 1 in patient 1 with the other 16 as well. So this also indicates that the deletions do have an effect with ASD. And they found that the deletion was not shown in the mother, but she carried the normal chromosome. And the last was that out of 13 patients, 10 of them did have the ataxin 1.
So our future outlook, in this experiment, the purpose was that they wanted to find out if CHIP and it can interact with wild-type axin 1 protein with and without the polyQ expansion. So CHIP is C-terminus of HSC70 interacting protein, and it's an E3 ligase that links protein folding machinery to a ubiquitin proteasome system. and CHIP in protecting from axin 1 induced neurodegeneration is what they were hoping to discover from this experiment. So also promotes ubiquitination of expanded axin 1. So ubiquitination deals in post-translational modification of cell proteins by marking them for degradation. So the method they did for this experiment was for determining axin 1 ubiquitination in vivo, the cells were transfected with axin 1 vectors that were then tagged, and 3 variants of human CHIP and hemagglutinine tagged ubiquitin. So the cells then underwent a treatment and were lysed with this anti-flag conjugated gel, and then washed and blotted with different antibodies. So the cell culture was then immunofluorescence. The cells were analyzed through this laser microscopy. And then PCR, reverse transcriptase PCR was used, and cycle numbers were increased to allow for a linear range detections of transcriptions, and the SCA1 transcripts were amplified. So in figure 1, these were describing the anti-flag immunoprecipitation, and then CHIP was pulled down, they found, by F-axin 1 and 82Q and F-axin 1 2Q. This was to find out if CHIP and expanded axin 1 were actually interacting with each other. In B and D, the CHIP accumulated in the axin 1, and the arrows are where the NIs, the nuclear inclusions, formed in SCA1 cells. So figure 3 and figure 4 both were about, they tested them in Drosophila eyes to see if they would CHIP would suppress axin 1. So that was just showing that they do cause degeneration in Drosophila eyes. The axin 1 did. And then with CHIP, it was reduced when it was overexpressed. And that is similar things as well. So what they discovered was that CHIP protects against neurotoxicity caused by the expanded polyQ tracts, and it demonstrated the importance of the protein complex in polyglutamine-induced neurodegeneration. So this is a very powerful genetic suppressor for this kind of neurotoxicity against it, and it could be used for treatment of SCA1 patients, Huntington, and accept more diseases as well. So the interaction with CHIP and axin 1 protein supported the idea that axin 1's tendency to misfold and expand and unexpanded. So they tested it for both the expanded axin 1 and the unexpanded, and the tendency for CHIP to interact with those is very high. So an overexpression of CHIP in Drosophila and SCA1 showed decrease in toxicity and protein levels of the expanded and unexpanded axin 1, which is good. Yay! And then CHIP localized to the cytoplasm, not the nucleus, but was found in all the NIs that were present in the brain tissue of the autopsied person that had SCA1 that they used for the experiment. So, therefore, finding CHIPs places a role in the clearance of expanded axin 1 in humans, which is exciting to think about. And that is similar as well. Overexpression can help decrease these levels of toxicity, expanded in axin 1. And that is what we have for you.
Yes.
Thanks, Dr. Paper. That's
you.
axin 1, by the way.
And
we love puns.
yes.
Sweet. Bye. Okay. It has been recorded. I think that was good. Okay, I gotta say, it was long.
Yeah, how long was that? 24 minutes?
Yeah.
Wait, how long was it supposed to be?
Not that long. I think it's only supposed to be between like 13 and 17 minutes. So,
What if since we have 3 people?
honestly, I kind of like that. I think we should just submit it.
Should it be like we extended the time limit just because we had 3 people?
Maybe.
We each talked the amount of time that we were supposed to.
I mean, I could trim mine down. I feel
like I rambled a lot. Sorry.
Yours sounded good though.
What are you talking about?
No, you didn't.
You were good.